I find it fascinating, the juxtaposition, because we're going to go from the very, very, very global to the very, very, very small, simple question. And I think that one of the things that we should do when we're looking forward is we should also look back. Because the basic information about behavior, which is fundamental to how the animal works, and also physiology, which is equally fundamental, we have kind of made these assumptions that all that was done 40, 50, 30 years ago, and we know about the behavior and the physiology of our animals, and we can start to do experiments, and we can start to do comparisons. And the answer is no. We've got huge, huge gaps in terms of our information about fundamental things like the behavior of the animals. And this is a very small study, but it's a look back and a comparison to look forward. And our question was, well, the cuttlefish have what people think of as a fixed and stereotyped action when they're catching prey. But the work was done by Wells in, I think, 1954, and then by Messenger in 1968. And everybody said, okay, it's a fixed sequence. Now we can go on to all the other things that we're worried about. And we said, well, is it a fixed sequence? And that's what I'm going to answer. Why bother? Sure, behavior is really general. Anybody can do behavior, that's actually true. You don't necessarily need a lot of fancy lab equipment. You can actually just go out on the shore. Why bother? Well, there's two reasons. Behavior is going to give us information about the physiology, information about the taxonomy, information about all the other aspects of the animals. Not only is it going to give us the fundamentals of the basis of what's going on with the animal, but it's very, very important when we start getting into aquaculture. If we're going to culture animals, we have to know what they're doing. We have to know what conditions they like. We have to know what conditions are favorable. We have to know. I was actually doing a proposal for some research I'm doing after I get here in Italy, and I submitted a proposal to the Institutional Animal Use Committee, and one of the things they said to me is, shouldn't you be giving your octopuses socialization? No, they'll kill each other, but people don't know, okay? So all these things that we want to do, first of all, we have to know the basic behavior, the basic physiology. And that's what we don't necessarily know. Now, one thing I have learned from many years studying cephalopod behavior is that cephalopods are learning machines, okay? So even before they're hatched, they're learning about the general substrate, they're learning about the potential prey. They're learning if they're making the eye dots. They learn that they only need to make them to visual predators. It's no use making them to a predator that isn't visual, and they don't. So they've learned this. They're learning about identity of different areas and species and interactions as adults. But an animal can't be just learning. It has to be having some fixed behavior, some guidance of learning. And we need to know the combination, and frankly, we don't. But we're getting there. Okay, so when we see, and I remember when I first saw a fixed sequence of behavior, I was in Ben Hills many years ago, and I saw the sand digging, and I thought, oh, good, a fixed action pattern in a cephalopod. I never see fixity. Let me go study it. And so one of the things I did is look at the limitations, particularly differences in the environment, which change the sequence of behavior. So if you're going to look at an action like the fairly fixed predatory sequence, then the questions that I can think of are, what's the situation that causes it? What particular environmental variables influence them? And how fixed are these actions? And why have we kind of taken the assumption that they are fixed? This is not a terribly good sequence, because what Messenger showed quite clearly and very nicely experimentally in 1968 is that there were three parts to this predatory sequence. The first would be a positioning which was getting the angular deviation with relation to the prey, straight on. The second one was the positioning, and that the cuttlefish would come to a fixed distance. Sometimes if the potential prey was too close, the cuttlefish would back up, okay? And then there was the strike. And I guess it's decided it doesn't want to play anymore. Okay, so this is an interesting example as well. And we'll be talking about ethical behavior and limitations on experimentation, and the 3 R's, which Giovanna could probably recite: reduce, replace, and reuse. And this is very important in research. And we're going to...
Not reuse.
Yeah. I should explain reuse?
No,
that's
not reuse.
No, okay. So we were doing a study in Italy in 2011, I think it was, and we were very interested in personality of cuttlefish. And so we had 19 animals in captivity, and we had 3 different conditions that we exposed the animal to. We gave them a threat. Sorry, first of all, we gave them alerting. We simply leaned over and looked at them. Then we gave them a threat, which would touch them on the side with a piece of wood or a brush. And then we gave them a feeding situation. And then we looked to see what the individual differences were amongst the animals in these situations. It's very interesting because, you know, for years I was doing experimentation, and you say, okay, there's a control system group, there's an experimental group. But with this one, you put all the animals through all the situations, and you say, okay, everybody had all the different situations, how do they vary? And while we were doing this, we took video clips. So each animal was exposed, it was given the same test each day for 10 days. And one of the tests was feeding. But we had them videotaped. And Claudio Carere, my associate, said, wait a minute, now we have all this videotape. Can we do something with it for a different study? And we said, well, you know, everybody assumes that the predation sequence is fixed, that there's no variation. But we have this video, so we found Francesca, a student, to take a look at the tapes and to figure out to what extent fixed really meant fixed. Okay? And that's where all this comes from. And we looked at latencies and durations, and we looked at different color changes, and we looked at actions like raising the 2 dorsal arms. But let me start with the latencies and durations. It's a fairly fast action. And see the 2 different sequences, the attention sequence, the positioning sequence, and the seizure with the tentacles. Seizure with the tentacles is very quick. In fact, I suspect it's quicker than we could actually measure. But the latency is in the seconds for all these 3 stages, and then the duration is very quick. And when we looked to see if it was stereotyped, what we said was, well, let's look at the coefficient of variation. And the coefficient of variation is a very obscure test. It's the mean divided by the standard deviation times 100. And clearly, the more stereotyped the behavior, the smaller the coefficient of variation. And when we looked at this, and there's not an awful lot done on this. People have used coefficient of variation, courtship of ducks. And when I did the sandpicking, I looked at coefficient of variation to see to what extent the sequence was the same or different when I gave different environments. And you see that the latency's coefficient of variation is relatively high, which means there's a reasonable variation. The actual tentacle extension and seizure has the smallest variation. So we see there really is, it's not very fixed. That's the first thing we had to figure out. And then we looked at the success rate for the first capture and the second attempt, and we found that the success rate isn't all that good. And we have to look more at this because if you've got an aquarium and you have a hungry cuttlefish and you put in a fish, which is their normal prey, it ought to be completely successful. And we're going to have to go back over the tapes and figure out why the success rate wasn't 100%. It's just not that high. The idea that a smart cuttlefish out in the environment has a fish right in front of it and can't get it worries me. Of course, one of the things that happens, and there's a paper I read subsequently, is that what the fish will do is wait until the predator's in place for this final capture and then move. Okay? So that's a defensive behavior of the fish, and we need to go back and look and see how often that happened. So one source of variability is the duration of these times. And in fact, as soon as we looked at that, we saw very little variability in terms of the duration, but significant variability in terms of the latencies. And so the latency for the three phases sank quite nicely between the first and the third trial. And then it didn't get any lower. So what we have clearly here is something we've always talked about as a fixed sequence, and it's a fixed sequence in the sense that you go through one and then the other and then the other, but your latency decreases. So this is a clear learning of the situation. Not the duration, which is how long it takes you to do it, but the latency, how long it takes you to figure out what you've got to do. Okay? And if you look back at Wells in 1958, you discover he saw the same thing. So I think to some extent, we ended up thinking that it was a fixed pattern, even though the evidence was probably there that it was only partly fixed. Okay? Sometimes I think people do the research and people get a general impression from the papers, and then it moves into legends and everybody says, oh yeah, it's a fixed pattern. Well, sort of. But the other thing we wanted to look at, because clearly we had been looking at personality in the cuttlefish, is we wanted to look at whether there was a variability amongst individuals in terms of all the different behaviors that we measured. And we found that there was more variability between individuals than within. Okay. And because we've been doing factor analysis to look at personality of the animals, we again did a principal components analysis and said, okay, to do a principal components analysis, basically what you do is you take all your measures and then you look for the correlations amongst them, and you look for a cluster of particular behaviors. And this is a subjective step, there's no question about it. You look at it and you say, okay, an animal has this and this and this. What would we call the general trend? And when I first did this with octopuses, I found three dimensions that I called activity, reactivity, and avoidance, and that predicted a lot of, a little bit over half of the variation amongst individuals. So this is what we did here, and we said, okay, given the variation in all these behaviors, can we find variations that cluster together, and then can we decide what they probably predict the animal is like? So activity is clearly an animal is moving around a lot. Reactivity, I had a reactive octopus once, and I was trying to test this particular individual in a round aquarium, and I'd bring it from its home tank, bring it to this testing area, and it would swim in circles all the way around, and it never settled down so we could ever test it. So there can be huge differences. The first dimension we found, which actually predicts 53% of the variance, which is quite a bit, looked to us like predicting that the animal would get on with doing the predation. It would be short latencies, it would be short durations. It seemed to be that this prediction was, it was an animal that was fast about doing everything, okay? And then the second dimension, which predicted 16% of the variance, seemed to be predicting to some extent whether the animal was focusing on whatever the task was, like focusing on the threat, when we were doing the threat test are focusing on, okay, I need to get the animal. And the third dimension was one that we called reactivity, and a reactive animal just ultra sensitive to all the environmental stimuli. And when we looked at it, we found that the first and the second dimension correlated with success rate in capturing the fish. So I'll go back. Okay, so the speed and attention correlated with actually catching the fish the first time. And dimension number 3, which was interesting because we were trying to figure it out, correlated negatively with hunting time. So that means that the more the animal loaded on that dimension, the reactivity, the less time it spent hunting. Not sure exactly how to interpret that. And when we looked, again, looking at the active individuals when they're hunting, and we looked to compare them with the threat and attention tests, we found that if they were more responsive to the alert and threat, they took longer attention time when they had the fish, and fewer attacks to success. So it looks as if they were more sensitive to the environment around them and less focusing when it came to actually getting the fish. And what this begins to tell us is that we should be careful not to say, oh, we have a stereotyped action sequence, because we don't. Even the ones that look as if they're fairly stereotyped, there's quite a bit of variability buried inside them. Fixed action pattern we don't use pretty well anymore. We now talk about a modal action pattern because there is so much variability. We want to figure out the average behavior, the most likely, because it really isn't fixed. And the other thing that we didn't have a chance to look at, but is very clear, is that you've got the attention, the positioning, and the attack with the tentacle extension. And both of those first two behavior clusters are closed loop in the sense that the animal continues to get sensory information while it's planning the attack. It's only with the tentacle extension, which is very fast, that it's open loop. And in fact, a fish that wanted to avoid cuttlefish would wait until just the beginning of the tentacle extension and then be gone. And the cuttlefish would not be able to pick up information between beginning the tentacle extension and actually either catching the fish or missing. So the actual take-home answer here is fixed isn't very fixed. And yet, when it comes right down to it, I keep thinking of it overall, you can't possibly have an animal that learns everything. It has to be the case that there's a basis of the kinds of things, the kinds of behaviors that the animal does, and that the small things change. So we really have, there's no doubt about it, in cuttlefish we have lifespan developmental learning. I don't see Aunt Sophie, but she knows. We have task-specific short-term learning, that's what we just saw here, and we have enduring individual differences. So when it comes right down to it, on the basis of relatively fixed behavior, I'm still convinced that cephalopods are learning machines. But we can figure out how we constrain the learning, when the learning is, and how the variation happens. And I would argue that the closer you look to finding out what the animal's doing, the more you see. And we should be doing much more close looking. Questions?